What are the basic components of a gene? We begin with a double-stranded DNA molecule. That includes a 5' to 3' strand and the anti-parallel 3' to 5' strand. I will represent both strands of the DNA with a simple line, and any sequence features I add to my model will represent the sequence that is found in the coding strand, which runs 5' to 3'. There are 3 basic gene components we want you to learn about today. The first one is called an open reading frame. Sometimes we call this an ORF or an ORF, and that simply stands for open reading frame. This is the nucleotide sequence that literally codes for the protein. We'll talk more about the components of an open reading frame in our video about translation, but it essentially contains multiple 3-nucleotide codons, and each codon codes for a particular amino acid. The second element you need to know about is the sequence element that initiates transcription. This is called the promoter. And note that it's upstream of or before an open reading frame. This is important as mRNA synthesis should begin before the sequence that codes the protein. The third element is the terminator, which is located after the open reading frame and causes termination of mRNA synthesis. Let's add some more details to our understanding of an ORF. An open reading frame is made up of many codons, and each codon codes for an amino acid. There are certain types of codons that will begin and end an open reading frame. The first codon of an ORF is called a start codon, and it always codes for the amino acid methionine. The most typical start codon that you will see in all 3 domains of life is an ATG, but there are other start codons in prokaryotes, and we'll talk about those more later. Each codon that follows codes for a different amino acid and determines the protein sequence. The last codon of the open reading frame is a stop codon, which does not code for an amino acid and signals the end of translation and the end of the protein. There are 3 different stop codon sequences: TAG, TGA, and TAA. Because each 3-nucleotide codon represents a single amino acid, the length of an open reading frame is always a multiple of 3 nucleotides. For example, a protein that is 30 amino acids in length will be coded by an open reading frame that is 93 nucleotides long. 90 nucleotides that make up the 30 codons that code for amino acids, plus the 3 nucleotides of the stop codon, which do not code for an amino acid. The next thing to know about the open reading frame is the double-stranded DNA molecule is that the code for the protein is in the coding strand. This is the strand that, when read in the 5' to 3' direction, has the exact same code found in the mRNA molecule, which is also read in the 5' to 3' direction. To synthesize an mRNA molecule that matches the same code as the DNA coding strand, the template strand of the DNA must be used to make the mRNA. The template strand runs in the 3' to 5' direction, and it is complementary in sequence to the coding strand. One final thing to think about: bacteria often code multiple proteins on a single mRNA molecule. That means that a single promoter initiates transcription of an mRNA molecule that contains multiple open reading frames. This is called an operon. Starting with the simple gene model we presented in this video, try creating your own model of an operon with 3 open reading frames.